So thank you for everyone for coming along. I'm going to speak for just a short, a little bit over 20 minutes, give a background and show some of the work we do, but also some of the thinking behind it and how we've interacted with industry partners, which at the time, maybe 10 or 15 years ago, were not traditional interactions. And thankfully, they're becoming more common and the interactions are much more integrated nowadays. This is a slide that one can show in many formats, but this is just the data from the FDA in the USA as reported in Nature Reviews Drug Discovery over the past few years. And it shows the approved drugs. And you can see that of all the approved drugs from 2017, 2018 and 2019, the proportion within psychiatry is vanishingly small. It's very low indeed. So, I mean, this emphasizes a major problem already that drug discovery within psychiatry does not really produce many successes, or at least currently it doesn't produce many successes. But also, you know, we have to think of the background here. You know, it's a whole other talk to try and unpick and unpack why these numbers of successes are so low. But one thing to note is that over the past 20 or maybe just slightly over 20 years, there's been a disinvestment in psychiatry R&D and drug development. And I'd list some of the major companies that stand out. There are definitely more as well, but these are the major players who have either no or very little investment in R&D in psychiatry. Some of these companies have also disinvested from neuroscience R&D in general as well, which is obviously indicative of a wider issue. So, you know, where does academic collaboration come in? There are many reasons why, say, myself as an academic, I'll speak from my perspective, might want to be collaborating with industrial partners, and many reasons why they might want to collaborate with us. Now, this is not an exhaustive list, but it covers many of the reasons that we have actually collaborated with industrial partners, and it covers a lot of the work we've been doing in the sort of translational space. So one reason actually is quite methodological. So understanding biological mechanisms of disorders. You'd have heard, if you've been to the meeting yesterday, you'd have heard a lot of this, say, from Amit Etkin's work on trying to look at large groups of patients with depression and seeing if there are subtypes, neuroimaging subtypes, for example. You might have candidate biological mechanisms. So a classical mechanism, say, in depression would be serotonergic system abnormalities. And there was a lot of research in the late 90s, early noughties, trying to understand if there were increases or decreases in serotonin receptor levels. So this is validating mechanisms. Pharmaceutical companies can often get together to form competitive consortium. This is where their drugs are not being used, but they recognize the need to accelerate research in a promising area. They might look at the research of a particular group of researchers and say, right, we want to be putting our drug into their research studies, but really we need them to accelerate and do the validation studies so that they're ready within a year or so. And that can be a real boon for a research group, because it's about identifying an approach, about your approach being identified, and being pushed forward. One thing that surprised me when I started working in this area is how interested the scientists within companies were in methodological development. So we've had investment. Some of our early retest studies, which we were doing 10, 15 years ago, actually were partly supported by grants from pharmaceutical companies that are very hard to get from traditional government funding agencies. It does sound quite boring looking at validation, but critically important to understand the reliability of the methods that we use. And when a technique's been around for 15, 20 years already, trying to convince a funder that no one's really done sufficient work in this basic validation is quite difficult. But we were very lucky in that some of our friends in the pharmaceutical industry got it straight away and were very pleased to fund it. They might be interested in accessing technology. We've got MRI scanners, PET scanners, so that speaks for itself, but also access to patients and patient records to do, obviously totally anonymized searches to try and understand what the needs are, where the problems are, what the successes and failures are, but also access to patients for the actual research studies themselves is critical. We do a lot of work on co-development of projects. We also can be sharing data to assist in deciding on the developmental pathway of a compound. We're designing a study at the moment where the animal research shows that the compound of interest is very successful in modifying cognitive function across many domains, actually. And we're designing a study where in humans, healthy humans, to try and look at the brain circuits that might be modulated to help with some of the decision making. So it's interesting to us as a neuroscience question. So we get to study systems that haven't been looked at because there aren't safe drugs in humans. And it's interesting to the company because, of course, they get some data that could potentially help address their questions internally. And of course, we can actually run trials, which of course seems like a funny thing to put at the very end. But of course, we can run early phase trials to get early indicators of efficacy. So I'll give a couple of examples. I'll base my talk around a couple of examples and a couple of studies that we've done. So you can get a flavour how we can fit into this space to help these critical decisions to advance our thinking in novel drug treatment development. So the example one is depression. So I won't introduce depression as a disorder, but it is a highly debilitating disorder. One of the major debilitating disorders, very high levels of morbidity, according to the WHO internationally. And the mainstay of pharmacological treatment is serotonergic antidepressants. I'm not dismissing other treatments as well. I'm just talking about pharmacological treatments here. I'm going to refer just to one trial, which is a large trial done by the National Institutes of Mental Health, which is called the STAR-D trial. I don't think I put it on the slide. I do apologise. And what this study showed is a large study looking at treatment responsiveness and efficacy. And we know that there is efficacy, but we know that it's imperfect. And we know that many patients don't respond well. But there are some numbers here from this study which really do highlight clearly that we need better, more creative, more advanced treatments for many patients. So 30% of patients initially remit, which is good. Okay, but 30% also have non-response after a year. Okay, and this group are often tried on multiple treatments, and we know that a lot of these patients will become what we call treatment resistant. It can take a while for remission. Arguably, 2 months is too long. And even if you do respond, there is a high possibility of relapse. So we have a treatment that works in some people for some of the time, but the response rates are not high enough, and the relapse rates really could be improved. So there's room, of course, for improved research in this area and novel treatments. The second example is schizophrenia. And the drug treatments for schizophrenia were all developed from the 50s to the very early 70s. There's very little new in this area. Similarly for depression, with the exception of ketamine, and you'll hear from Katia as well about psychedelics. So you've got cognitive deficits in schizophrenia, which are a major symptom. Most domains are affected. They actually predict functional outcome very well, much better than positive or negative symptoms, and there's no licensed treatment. And existing treatments don't really knock cognitive function very much, don't really improve it very much. We've also known for a very long time that glutamate abnormalities are associated with schizophrenia, yet there's no licensed glutamatergic treatment as well. So I'm just going to expand on the schizophrenia and glutamate story. So this starts back in the very late 50s when PCP, phencyclidine, was shown to exacerbate symptoms of schizophrenia in patients, but also to some extent produce similar symptoms in healthy volunteers. And these 9 patients and 9 controls are shown on the right against a range of states that they self-reported. Okay. But at the top right, you see phencyclidine. But actually, phencyclidine is not a great drug. It's very potent, not very nice. It's not as safe as one of its follow-on drugs, ketamine. So the same company, Park Davis, produced phencyclidine, and a couple of drugs later were ketamine. But they were looking for anesthetics as well. And ketamine is a good anesthetic. But when you give it in lower doses, the effects seem to produce something we call psychotomimetic effects. So the NMDA receptor is shown here, and ketamine blocks the open channel. And by blocking the open channel, the little cartoon on the right shows that it disinhibits the GABAergic system, GABAergic system's effects on the glutamate system, and can promote glutamate release in this very simplified diagram here. So is it a model of schizophrenia? Okay, so you've got this NMDA hypofunction. Oops. You've got this NMDA hypofunction, which leads to disrupted cortical activity, reduced GABA release, and increased glutamate levels. So if you give individuals, healthy volunteers, the same scales that you give patients, then you can actually score in the same range as patients. So that led to thinking that actually this is a model of schizophrenia itself. Now, we no longer think it's a model of schizophrenia per se, which is way more complex than enhanced glutamate release and disrupted glutamate transmission. But with regards to the glutamatergic abnormalities you see in schizophrenia, well, it's a fantastic way. What a fantastic way to study that component of a disorder. If you can use a single drug to mimic the glutamatergic abnormalities, then it's a great way to study the glutamatergic abnormalities in studies which can be run much faster than in patients and without some of the complexities of, for example, long-term treatment with other medications. Okay, so ketamine is a model of glutamate dysfunction, which is relevant to schizophrenia. And so this is one of our studies shown in the 2 graphs, A and B in the middle. But first of all, we need to validate that ketamine at relatively low doses actually does increase cortical glutamate levels. And there's intracortical recordings in animals, magnetic resonance spectroscopy in rodents. We did an MRS study in humans, which is shown in the graph. And what we showed was actually the psychotic-like symptoms people were reporting was actually correlated to the glutamate levels. And there are more recently some more sophisticated studies using 13C MRS, which actually suggests that it's the glutamate turnover rather than just glutamate content that matters. So it's actually glutamate release. So how do we then use this to look at novel drugs? Well, you can give ketamine to obviously create this model, but instead of the behavior, which does occur, of course, but it is reasonably robust it's reasonably reliable, but everyone has different experiences. And if you want to study the experiences, you need to use quite large samples. But what we found is that when you put people in the scanner and you inject people or infuse people with ketamine, and you apply this model, this thick black line is from some pilot data and data from another group showing the time course of the effects of ketamine on brain function, and then you model things like head movement, for example, then we can see quite a reliable signal in the brain, mostly increasing activity due to the ketamine infusion, and it's reliable. So we can see that over multiple sessions. We thought we might get rapid tolerance, but you don't. And we can do multiple doses. And we can show that this translates nicely from animals to humans. And in the rodent studies, of course, what they can do very easily, or relatively easily, is put in drugs to block the effects of ketamine, to try and validate that they are picking up glutamate modulation at a level that is relevant to studying other drugs. And they're able to show that that a D2 antagonist, a pure antagonist like raclopride, which theoretically should not affect the influence of ketamine, doesn't have an effect. But drugs like clozapine, which can modulate glutamate through 5-HT2A receptors, lamotrigine through a different mechanism through sodium channels mainly, and even novel drugs, MR2/3 agonists, can modulate the ketamine effect. So we wanted to translate that into humans. So we've got pH in this red box here. This is the model overall for our schizophrenia research, where fMRI tasks can be developed in healthy humans and tested in patients. But the example I'm giving you today is when we're tracking, say, the ketamine response in rodents, and we're trying to replicate this in humans, and we can add in other drugs. So here's the study design. We used a similar drug to the animals, lamotrigine. We used risperidone, not raclopride, in the humans. Risperidone is more interesting because it's more commonly prescribed and it has this 5-HT2A effects as well. And we used healthy volunteers. And we were able to show, I won't show all the results in this talk, but we were able to show very clearly that lamotrigine and risperidone both reduced the ketamine-induced pattern of changes in activity in the brain very robustly in terms of brain activity and brain connectivity. So really validating the methodology works. And then we can put in novel drugs. So we put in 2 drugs from Eli Lilly, and we were able to look at different doses, and we showed very clearly that the low and the medium doses of both compounds had very little or no effect, no detectable effect, but the highest doses had a very clear effect. And also, some of the delusions produced by ketamine were also attenuated, but only by the highest doses. So it tells you need to push this system quite hard to see behavioral effects. But we validated it against this ketamine model, which had only been done in rodents before, but really validation in the species you want to be treating is important. So, what are the other reasons? What are the general reasons you might be using this sort of neuroimaging technique? Well, when you're discovering a disorder, discovering the brain systems that might be involved, you can define the brain systems. Obviously, you might find that they fractionate with different patient subgroups, showing difficulties in different systems, or you might find fractionations across symptoms. So some symptoms are associated with some brain areas, whereas other symptoms are associated with other brain areas and networks. And of course, you can replicate this work. You have major challenges in diagnostic heterogeneity, definitions of the processes, and heterogeneity in the patients as well in terms of their previous experiences and current experiences. You can use these methods to validate your brain systems you're looking at. So for example, you might show that reward system impairments come up again and again. Okay, they might not overlap one-to-one with diagnosis, but they might be a great target for testing your theories. So you can validate them. You can look at existing drugs, and you can look at variability in terms of those people who respond and don't respond. And again, you can look at processes, different tasks, different brain networks, mediation analyses to see if they really do mediate the symptoms as well. And also you can look at testing novel compounds and trying to predict which novel compounds are going to be more successful. So in depression, the candidates that have been mostly used are looking at emotional responses to faces and altered responses to rewarding stimuli. And the example I'm going to give you next is with ketamine in reward systems. So we know that ketamine has emerged as an antidepressant, a rapidly acting antidepressant that has a license for treatment-resistant depression. And it's a very interesting drug because I've told you already about the psychotomimetic effects you get in the acute phase, which this cartoon shows coming up and down, and they've gone within a couple of hours. And we've done imaging at this acute phase and this slightly later phase, and you can see brain responses very clearly. There have been quite a lot of studies in patients looking at delayed responses, so 24-hour when the antidepressant responses tend to peak, showing connectivity changes between key brain circuits, and also some changes in connectivity which relate to the early subjective and glutamate effects here, which is fantastic. And also there are effects beyond the delayed effects of 24 hours into 2 days or even over a week as well. But there are small studies, so we're really waiting desperately for replication so we can understand what systems are most robustly changed and what systems most robustly underpin the longer term effects, and maybe we can even predict who might respond better or worse. We know very little about this early phase, 2 hours in, when you start to get the emergence of antidepressant effects, but the psychotomimetic effects have gone. And that might give us some clues as to the cognitive processes which might be mediating these later effects. So the study we did was looking at reward processing in this 2-hour window as a way of understanding, potentially understanding anhedonia, which is the impairment, of course, that might be improved. There are plenty of studies looking at reward systems in depression. So we did this study, which we called Karma, and we looked at rumination and reward. We actually had remitted patients in this study because we wanted unmedicated patients and to focus on the effects of ketamine and not the effects of ketamine secondary to symptom change. It's about validating the mechanisms or the processes in the brain at least. And we used a task called the MID task, which we can easily scan 2 hours after the ketamine infusion. And we aim to see if we could see changes in those systems. And there are a number of studies at the bottom which justify looking at this time window and these sorts of processes. So if you don't know this task, it's a reward prediction task where you have a target you respond to as fast as you can. It's preceded by a cue, and that cue might signal a potential for reward. And we're interested in this period of time here while people are waiting for the cue, this anticipation period responding to the cue, and this outcome period when they actually get a reward or don't get a reward. I've just got a few slides left. And this study is in submission, so it's not published yet. I'm very happy to share it with you. And what we showed in this 2-hour window, when you have no obvious effects of ketamine subjectively, is across the reward network, which we defined here as these multiple regions that respond to the reward anticipation and receipt, that when you anticipate a reward across the entire network, you can see the orange bars, which are ketamine, are consistently higher than the blue bars, which are placebo. We actually get a main effect of ketamine, but the strongest effect is within the dorsal striatum of the caudate part of the dorsal striatum as well. Actually, we do get a main effect across the entire reward system. And during the feedback, so when people find out they've won a reward, again, it's more obvious here, but we get an increase in ketamine across the entire network here. It's particularly strong in a few regions, but again, we get a main effect across the entire network. And in fact, it's strongest for the low wind trials. What it seems to be doing is making the low wind trials look a bit more like high wind trials. And what's really interesting is that the caudate response and the ventral tegmentum response, so really key dopaminergic nodes, they correlate with one of the metabolites of ketamine, which has been called 2R6R HNK. They correlate with the concurrent metabolite levels, and in animal studies and some emerging human studies, this has been proposed as a key mediator of the antidepressant effects, the metabolite of ketamine, hydroxynorketamine, which acts at AMPA receptors as well. So this is a very interesting result, and it shows the power of the methodology. And you can see the levels here. So now we can fill in one of the gaps in our knowledge here. We know at least that we can get this early modulation of reward processing. And of course, what we'd like to do now is to track that over time. So every study I've shown you is selected for the presentation today were collaborations with industry. So even those retest studies with ketamine, understanding the effects of ketamine against laboratory and trying to replicate these animal studies. We also did quite a bit of development of methodology, was supported in part by Pharmaceutical, by Eli Lilly, who of course had a very strong interest because they had their mGlu2 program running at the time. And these depression studies we've been done recently were supported by like a stimulus grant, I suppose, an investigator-led sort of stimulus grant from Johnson & Johnson. And I must say, they've both been great partners, very good scientists within the company who've done these sorts of studies. And we were free to publish and also share our data with other people as well, which is important. I just want to finish by saying that when we're doing these pharmacological imaging studies, we have a multitude of approaches available to us. I talked about fMRI with tasks and some resting and some PHMRI here. Actually, I didn't talk about resting state and CBF, but in all these areas, we've actually benefited from collaboration with pharmaceutical companies in developing some of the methods for pharmacological imaging, and also got to test some novel mechanisms which to date have only ever been tested in animals. So there's some real advantages to this type of work. And there are always people to thank, and there are also other people that fund our research. So thank you very much for your attention.